Hi guys, I'm here to talk a little bit about evidence of common ancestry. I'm going back a week. Evidence of common ancestry, and then we'll talk a little bit about molecular clocks and tree of life. All right, so the first thing is I can describe examples of processes and features that are found in all organisms. In other words, they are core and they are conserved in all organisms. And so the first place I want to start are those characteristics of life. So all living organisms are made of cells. They reproduce, they grow and develop, they maintain homeostasis, respond to the environment, contain genetic information, obtain and use energy, and evolve. 8 characteristics of life. Why do they do or have all 8 things? Because their ancestors did. And why do their ancestors? Because their ancestors did. And so what we can do is we can trace that back to the very first ancestor that was able to do all 8 of those things, and therefore passed it down to all of us. If you want to be more specific, when we look at contain genetic information, we all have DNA and RNA. Our DNA and RNA is made of A's, T's, C's, G's, and RNA has U's. We use the same code, that first base, second base, third base codon wheel or codon chart. We all use that same code to translate our RNA to our proteins. So when we're building proteins, we're using the same code. We use the same RNAs to do the same things. mRNA is used as the code for translation. tRNA helps build by bringing in amino acids. rRNA makes up the ribosomes. So we have these things that are structurally the same as far as our genetics go. Other things that we have in common: the enzymes that we use, so the polymerases, we have RNA polymerase for transcription and DNA polymerase for replication. We can stop transcription with repressors, all those kinds of things. There's that code, the anticodon-codon matching, so all of that goes into our genetics. Another one of our characteristics of life is that we can obtain and use energy. We all have a way to do that. Now, plants do it differently than animals, right? Animals must consume things. Plants are able to do photosynthesis. They're able to produce their energy through the process of photosynthesis. So we have different ways of doing it, but we all have a way to do it. And so originally, the concept of capturing and storing and using energy was in that ancestor, but then through speciation and diversification, through divergent evolution, we started to see different ways of doing that. Okay, so remember, evolution talks about the unity of all things. We have a common ancestor, as well as the diversity that we, through divergent evolution or adaptive radiation, we have gotten all this diversity in species so that we can all fill different niches or niches. Maintain homeostasis is a big one, and we have to maintain homeostasis in all kinds of different ways. Water is one of the biggest ways. We'll talk about that in a second. But gas exchange, anything that does aerobic cellular respiration is going to need oxygen, and it's going to produce CO2, including plants. Yes, through photosynthesis, they produce oxygen and use CO2, but they also do respiration. And so we have to have ways to get those gases in and out of our bodies as needed. And everything from a little old bacteria to a big old cow does this. Even anaerobic, they don't need oxygen, but they do still produce gases and things that they need to be able to get rid of. Digestion, we need to be able to break down our food. Again, even plants. Plants make sugar through the process of photosynthesis, but then they have to turn around and break it down through respiration. So everything has to be able to break down that food, whatever that food looks like, in order to use it. And so we have different ways of doing that, but we all are able to do it, and that again comes from a common ancestor. Waste production and elimination. In doing these chemical reactions, we are producing waste, and waste can be anything from urine to feces to carbon dioxide. Carbon dioxide is considered a waste product. Oxygen is considered a waste product when you're talking about photosynthesis. So how do we get rid of those things so that they don't build up and toxify our body? And so again, we all do it a little differently, but the fact that we all have a way to get rid of our waste tells us that our common ancestor gave us that. And then those methods of getting rid that are different are through adaptive radiation. Osmoregulation, that's the big one with water, maintaining the correct water balance. Dehydration or overhydration, hyperhydration can both be fatal, whether you're a single-celled organism or a multicellular organism like humans. And so we have to have ways to maintain that water balance, and we do. Again, the methods are different, but the concept of maintaining that water balance is the same. Came from a common ancestor. And then we all have a way to move things around our body. And so we can be super simple. Plants, and well, not, yeah, I guess plants do too, but I know things like euglena, which is a protist, they do cytoplasmic streaming. So the cytoplasm literally, you can go online on YouTube and Google, or YouTube, Is it called Google when you're on YouTube? Anyway, cytoplasmic streaming, and it'll show you a video, like a few seconds. They're not long videos, but it shows the cytoplasm moving and you can see the chloroplast moving. And so that's just a way to make sure their nutrients are kind of getting stirred up so that their sugar isn't all in one place and their oxygen's in another or whatever. You want to make sure it's all kind of everywhere. And so even single-celled organisms have a way to move things around their cell. Of course, as a multicellular organism, we have ways to move things around as well. Maximum parsimony is the next one. I can define maximum parsimony. So when we do phylogenetic trees, remember it represents a hypothesis, and so sometimes there's a couple of different ways we could interpret this. We can say 3 broke off first, or nope, 2 broke off first, or nope, 1 broke off first. And so then what we do is we look at our evidence for speciation, in this case it's DNA, and we look at how many changes or how many things had to happen, how many events, evolutionary events had to occur in order to get each of these trees. And so when we look at the first tree, it took 6 events for that to occur. And the second and third hypotheses or phylogenetic trees, it took 7. So we're going to assume the simplest one is the most likely one, and so we're going to assume that that is the correct one. It's not always right, but it's kind of a concept that we use that's generally right, because if evolution had to make 7 changes versus 6, 6 is more likely. Parsimony means stinginess, and so it's the maximum stinginess. I'm going to be really stingy with how many changes I'm going to say happened, and I'm going to go with 6 rather than 7. So that's the concept of maximum parsimony. And then the next thing, because we got a tree of life too, is I can explain the concept of a molecular clock. A molecular clock is just a tool that we use to enable us to figure out when 2 species evolved. And so now I'm going to switch over to an example. So let's say that we have these 4 species, and these could be like 4 species of monkey. Like maybe this is a lemur and I can't think of a bunch of monkeys right now. Rhesus monkey, a macaque, and a gorilla. I don't know. I'm just making these up, okay? So in your mind, picture actual species. And what we know from evidence that we have, fossil records, whatever, is that species A and B split about 100 million years apart. And when we look at, let's say, their DNA, it's about 25% difference. Okay, and then species A and C, we know, broke apart 300 million years ago, and they have 75% difference. And species A and D broke apart 200 million years ago, and they have a 50% difference. And so what we're going to do is we're going to graph that. And so we're going to have, oops, I didn't put my time on here. We'll go with 100. I'm just kind of eyeballing it because I don't want to take the time to count it out. That would be a waste of your time. Okay, so 125. 250 and 375, okay? And then I'm just going to connect those lines as best I can without a ruler. Okay? And technically you don't go past the last points, but in case I need to extrapolate, I will, but I just know that these are guesses. Of course, so is the interpolation. All right, so things that I might get asked is, what is the molecular clock? What's the rate of mutation? Well, rate is slope. And if it doesn't tell me, I'm generally going to take the first and last, okay? And so I'm going to do 300 minus 100, and I know those go on the bottom because they're on my x-axis. Remember, when you do slope, which is just y2 minus y1 over x2 minus x1, the x goes on the bottom. And then I'm going to put on top 75 minus 25, so that's 50 over 200, which is 0.25. Okay, and so that would be my mutation rate. What that's saying is for every 100 million years, I'm getting about a 25% change. Okay. Something else, so that may be one thing they ask you, is to calculate the rate or to calibrate the clock. So that's what it means to calibrate the clock, is to come up with that rate, okay? Another thing they may ask you is they might say, well, what if we find a species and we look at their DNA and there's 32% divergence, okay? So 32-ish is going to be around that line. Boop, 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 boop. So what I'm going to do is I'm going to interpolate. So I'm going to take, let me do it actually in a different color. I'm going to take that 32% and I'm going to see where it would fit between 2 data points. And then I look down here and I don't know, I'm going to say it's not quite to 150, that's about 150-ish. It's above 125, so maybe, I don't know, 135, 140, somewhere in that range. So if it has 32% divergence, then I'm going to say that's about 135 to 140 million years ago that they diverged. So once you've calibrated that clock, you've graphed it, you've got the slope and all that, then you can do interpolation or extrapolation if you need to go past the points to figure out new species. So maybe this is species E, and when we compare species E to A, there was 32% divergence, and we want to know how long ago did they probably mutate or speciate. It was probably 135 to 140. So those are just some ways that they may ask you to use a molecular clock. I don't think they will, but if you have one, graph it, calculate the slope, interpolate, extrapolate, whatever. All right. So that says I can use a molecular clock to estimate the time of divergence between two species. You do that through interpolation or extrapolation. And then the last thing is tree of life. And so with the tree of life, it says I can explain the overall structure. So what we have is a common ancestor that's split into two species. One became all of the bacteria, all of the eubacteria. The other went in a different direction, which also split again, one branch becoming archaea domain and the other branch becoming the eukaryotes. So somewhere around this split, we saw endosymbiosis occur, okay? And so all of your eukaryotes, protists, fungi, plants, and animals are all up here. I can give some examples of structures that are common to all eukaryotes. So the first eukaryotic ancestor, which would have been somewhere on this line, had a cytoskeleton. They had membrane-bound organelles like the mitochondria, linear chromosomes. At some point, somehow our chromosomes got split from circular to linear, and endomembrane systems. So since all eukaryotes have this, we can assume that the first eukaryote did as well. And then I can define horizontal gene transfer and tell why it tangles the tree of life. So parent to child is considered vertical transfer. You got your DNA from your parents, that's vertical. We can trace that very easily. But there are other ways for you to get DNA. For example, a virus may pick up DNA from your sister. Well, that would still be a lot of your DNA, but let's say somebody in your class that you're not related to, and then you catch that virus because your classmate coughed or whatever, and now you have that virus. Potentially, you could get some of their DNA. Is it likely? No, but it can happen. And so that tangles up the tree of life because it makes things look related that actually are not. And so what we see is we see these organisms getting DNA, and it's creating these horizontal lines on the tree of life and making things look related that aren't because it wasn't a vertical transfer. Other ways this can happen are transposons, which are jumping genes, plasmids, which are extra chromosomal DNA that can go into bacteria but also into other things. There are plants that can take up plasmids as well. We've talked about viruses. Bacterial infections can do the same like viral, or something like endosymbiosis where two organisms fuse and live together. And so now that DNA looks like it's one organism, but it used to be two. And so that can create a idea of relatedness that actually isn't true. So that is the idea of horizontal gene transfer and how it can tangle the tree of life, and I hope that helps. If you have any questions about any of this, just let me know and I'll be happy to go over it with you. Thanks, guys.